Hello, I'm Lars Klimaszewski. I'm professor for neuroanatomy and head of the Department of Anatomy, Histology and Embryology at the Medical University of Innsbruck. I want to introduce you to the basic neuroanatomy of the human brain to help you understand the morphological basis of neurobiology and neurology. This course is also intended for those working in the Human Brain Project to familiarize themselves with structure-function relationships in the human central nervous system. During my tutorial, I will show about 30 slides. Some of them contain schematic drawings with the relevant structures labeled using the official anatomical nomenclature. The graphs have been taken from a compendium I wrote years ago for the MD and PhD students in Innsbruck. I'm grateful to my colleagues in the Division for Clinical Anatomy for providing images of frozen brain sections I'm going to show. In addition, I will demonstrate some histological specimens. They have been prepared at our Division of Histology. I will also show a brain model to demonstrate the spatial relationships of the various components of the brain. Let us now start with a short introduction into brain development. The entire nervous system is derived from the ectodermal cell layer of the human embryo, the neural plate, in a process called neurulation. 3 weeks after fertilization, the plate forms paired folds which unite to create the neural tube that will later form the medulla spinalis, better known as the spinal cord. The tube closes at the end of the first month of development and expands in the cranial region in the form of 3 vesicles. These vesicles are called prosencephalon or forebrain, mesencephalon or midbrain, and rhombencephalon or hindbrain. In the second month, the 5-vesicle stage appears. The prosencephalon now forms 2 telencephalic vesicles, the cerebral hemispheres, and a 3rd vesicle below in the midline, the diencephalon. The midbrain remains a single entity, but the rhombencephalon will divide into the metencephalon, later in development called pons and cerebellum, plus the myelencephalon, later called the medulla oblongata. The central canal inside the medulla spinalis enlarges cranially to form a diamond-shaped 4th ventricle, shown here, which communicates via a thin aqueduct located in the mesencephalon with the 3rd ventricle and with the 2 lateral ventricles that connect with the 3rd ventricle via the foramen of Monroe. All 5 ventricles contain the cerebrospinal fluid. A lateral view of the 5-vesicle stage reveals the developing hemispheres and the diencephalon with the Augenanlage, the optic cup. Note that the 5-vesicle stage reveals a brainstem that buckles as development proceeds. See the flexures at the level of the pons and at the beginning of the spinal cord. At a later stage, the embryo is now 60 days old and about 3 centimeters long. The cerebellum becomes visible. It develops from the dorsally located rhombencephalic lip. The isthmus forms between the midbrain and the rhombencephalon at a position where the cranial nerve number 4 exits the midbrain dorsally. The cranial nerves 5 to 8 are located in the upper rhombencephalon, while the nerves number 9 to 12 exit the medulla oblongata. Note that the olfactory nerve number 1 is part of the telencephalon. Both the 1st and 2nd cranial nerves are of central nervous system origin, whereas all other nerves are derivatives of the peripheral nervous system that originates alongside the neural tube from so-called neural crest cells. They differentiate into peripheral ganglia and produce all glial cells of the peripheral nervous system. The cranial and spinal nerves connect the brain and spinal cord with their sensory targets. with the musculature and with our inner organs. The spinal medulla maintains its segmental origin throughout adult life and gives rise to 8 cervical segments, 12 thoracic segments, 5 lumbar, and 5 sacral segments. Cranially, the medulla oblongata extends the spinal cord, followed by the pons, the midbrain, and all 3 of them, together with the cerebellum, form the brainstem. Tucked underneath the cerebrum in the posterior cranial fossa is the cerebellum that plays an important role in the motor control. In this median sagittal view, the central canal, the 4th ventricle, and the aqueduct are shown, and the diencephalon above the midbrain is part of the forebrain and contributes to the hypophysis, the pituitary gland that produces various hormones regulating our inner organs. And also in the posterior part, the pineal gland is found, alternatively called the epiphysis, which produces melatonin, a sleep-regulating hormone. The optic chiasm is shown here. It is the crossing site for some of the axons running in each optic nerve, our cranial nerve number 2. The corpus callosum is visible above the diencephalon, which is the major fiber commissure in the brain. It connects homologous regions of the neocortices in the 2 hemispheres, which cover nearly all of the brainstem and diencephalon. The telencephalic pallium develops into the frontal, parietal, temporal, and in the occipital lobe, as well as into a 5th lobe buried inside and behind the lateral sulcus, the insula. The insula forms a pivot around which the expanding hemispheres rotate in a Widerhorn-like manner during development. So the final form of the cerebral hemispheres is the result of millions of neuronal precursors migrating from the subventricular zone into the anterior rostral, the posterior occipital direction, as well as downwards and laterally to form the temporal lobe. Please note that the neocortex is folded, thereby forming sulci and intervening ridges. These are called gyrus or pleural gyri, which together produce a larger surface area in contrast to the smooth surface of smaller vertebrate brains that are called lissencephalic. In the human brain, 2/3 of the neocortical surface are hidden in the grooves. 2 major gyri are integrated here, the pre- and the post-central gyrus. They contain the primary somatomotor and somatosensory areas of our brain. Instead of following a descriptive approach for each component of the brain, as I'm planning to do in this tutorial mainly, teaching neuroanatomy might alternatively focus on the different functional systems. Following this approach, the central nervous system receives and integrates input from the various sensory systems and sends output signals to different effector tissues, mainly muscles and glands. First of all, there are 5 special sensory senses in our head: the auditory, vestibular, optic, gustatory, and olfactory systems. They feed information into the brain similar to the peripheral sensory input coming from the skin, the so-called exteroceptive sensation, from muscles, tendons, and joints, the proprioceptive systems, and from the inner organs, enteroception. Inside these different tissues, we find receptors for pain, the so-called nociceptors, for warm and cold sensation, the thermoceptors, for touch or pressure, the mechanoceptors, and for various chemicals and drugs, the chemoceptors. The brain and the spinal cord integrate these inputs to produce a behavioral response via the motor or efferent nerves, which are divided into a somatic and visceral component. The somatomotor system performs involuntary and voluntary movements via the skeletal and also the skeletal but called branchiogenic cranial musculature. It is innervated by cranial nerves number 5, 7, 9, and 10. Visceromotor nerves belong either to the parasympathetic or to the sympathetic system. They innervate the glands as well as cardiac and smooth muscle cells. The enteric neurons comprise a separate entity of the peripheral nervous system, but are extensively regulated by visceromotor and viscerosensory neurons. We will now start with a discussion of the spinal cord, including the main motor and sensory systems that connect the brain with the periphery. In low magnification of a histological H&E stained transverse section through the spinal cord, it is not easy to differentiate the gray matter containing the neurons from the white matter surrounding it. The gray matter contains the neurons, the nerve cells, and acquires a butterfly-like shape, exhibiting a ventral and a dorsal horn. The white matter is divided into the anterior, lateral, and posterior funiculi and contains the output processes of neurons, the axons, which project either upwards, cranially to relay afferent signals, or caudally to send afferent information downwards to the motor neurons. A larger magnification of the dorsal horn reveals a V-shaped mass of gelatinous neuroglia, termed the substantia gelatinosa, which contains small nerve cells and lots of nerve fibers, which are mainly non-myelinated. These fibers lack the fatty white substance that is typical for neuronal processes exhibiting fast conduction velocities by forming an electrically insulating layer of plasma membranes around the axons. Outside the dorsal horn, you see a large amount of delipidated, formerly myelinated nerve fibers, in addition to nuclei of the glial cells. The glial cells are the myelin-producing oligodendrocytes, as well as the astrocytes, the second major group of cells in the central nervous system. In the ventral horn, we observe large cell bodies. These are the motor neurons that are surrounded by neuropil consisting of neuronal processes and glia, mainly astrocytes here. In this schematic horizontal section through the upper cervical spinal cord, the dorsal and the ventral horn of the gray matter, as well as the intermediate zone, are indicated. The latter contains mostly autonomic neurons involved in sympathetic control of our inner organs. Broer-Rexed in the early 1950s identified 10 layers within the spinal gray matter in order to group neurons by their ability to process the same or similar modalities. Lamina 1 to 6 are located in the dorsal horn, while 7 and 10 comprise the intermediate zone. Lamina 9 indicates the nuclei of motor neurons projecting to somatic muscles, and 8 indicates the surrounding neuropil. On the left, I have labeled the descending fiber tracts inside the white matter. Of course, all fiber tracts are found bilaterally, but for didactic purposes, here are shown only on one side. The lateral corticospinal tract, which we will discuss in the next slide in more detail, is the functionally most relevant afferent fiber tract, with some fibers running in the anterior funiculus, as shown here. Other afferent tracts originate in the brainstem, for example, the tegmentospinal axons, located ventrally from the corticospinal tract. Some come from the diencephalon, like the autonomic fibers, innervating the visceromotor neurons located in the intermediate zone. Olivo, reticulo, vestibulo, and tectospinal tracts are important for maintaining stability and regulating posture and gait. On the other hand, afferent systems are shown on the right side and in the dorsal funiculus. Here we find the gracile and the cuneate fasciculus, which starts at thoracic segment 3. These fibers transmit mechano- and proprioceptive information to the brainstem, but are also heavily involved in intraspinal networks indicated by a large number of axon collaterals, which innervate different segments of the spinal cord. Intersegmental connections also run in bundles around the gray matter, shown here, and they are called fasciculi proprii. The posterolateral tract, which carries the incoming sensory axons from the dorsal root, is indicated here, named after Lissauer. Another important pathway transmitting pain is called the lateral spinothalamic tract. It is supplemented by an anterior part that is responsible for mechanosensitive impulses. The spinotactile, spino-vestibular, and spino-olivar connections provide highly relevant sensory information from muscles and joints, which is unconsciously processed and important for posture and gait. This is also true for the very laterally located spinocerebellar tracts, the anterior and the posterior one. Let us now look at the functionally most relevant tracts in more detail. The pyramidal or corticospinal tract represents the major motor pathway in the central nervous system of humans. The tract originates in various cortical areas, mainly in frontal regions like the motor and premotor cortices, in the frontal eye field, prefrontal cortex, in the speech center and cingulate gyrus, but also in the postcentral somatosensory gyrus. The corticospinal tract then passes through the posterior part of the internal capsule, which comprises all fibers running from the thalamus in the diencephalon upwards and all downwards projecting fibers from the cortex to the brainstem and spinal cord. The CST is located in the middle portion of the midbrain, in the peduncles, and descends through the basilar ventral part of the pons to the pyramid of the medulla oblongata, where most of the fibers cross to the other side. The tract then descends in the lateral funiculus before innervating motor neurons located in the ventral horn of the spinal cord, contralateral to the site of origin, since both tracts, the anterior and lateral one, have crossed the midline. In case the ventral horn is damaged, for example at cervical level C7 and C8, a flaccid atonic paralysis appears, an inability to move the hands. In contrast, if the corticospinal tract itself is affected, here with the lateral and the anterior part, the inability to move the muscles becomes spastic over time. That is, the muscle tone increases progressively, particularly in the anti-gravity muscles, the extensors of the joints. Now I want to discuss the major sensory pathways. The dorsal column, also called medial lemniscal projection, provides the parietal lobe of the brain with an instantaneous body image. It makes us aware of the position of our body parts at rest and during movement. Without this information, the execution of movements would be severely impaired. This pathway starts with mechanoceptors in the periphery, which are connected to primary sensory neurons located in the spinal ganglia, also called the dorsal root ganglia. There, centrally projecting axons ascend through the dorsal funiculus to the gracile and cuneate nuclei located in the dorsal medulla oblongata. From here, the second-order neurons cross to the other side and project in thick fiber bundles called the medial lemniscus to the contralateral thalamus, which is the major sensory relay center located in the diencephalon. At the level of the midbrain, the medial lemniscus is located between the spinothalamic tract, this small structure here, and the red nucleus, dorsal to the substantia nigra. These structures we will discuss later. The thalamic subnucleus containing the third-order neurons relevant here is the ventral posterolateral nucleus. The neighboring posteromedial nucleus serves the head region. It receives input from the trigeminal nuclei, the fibers of which ascend in the trigeminal lemniscus, shown here. The nucleus of the fifth cranial nerve receives its information mainly from the trigeminal ganglion, the counterpart of the dorsal root ganglia in the head region. The final and fourth neuron is located in the primary somatosensory cortex, the postcentral gyrus. The tactile representation is orderly arranged from the lower leg at the top to the facial regions at the bottom of the parietal lobe. The dorsal column syndrome, induced, for example, by a lesion at the level of the 10th thoracic segment, results in hyperesthesia, which refers to a reduced sense of touch, and in sensory ataxia, which indicates a lack of coordination of voluntary muscle movements resulting in severe gait abnormalities. In contrast to the dorsal column pathway, the pain and temperature pathway crosses the midline in the anterior commissure at all spinal segmental levels. With their first receptor neurons located in the spinal dorsal root ganglia, the centrally projecting neuroaxons reach the dorsal horn and branch into ascending and descending collaterals, forming the posterolateral tract of Lissauer, as mentioned before. After penetrating the dorsal horn, they synapse onto interneurons or directly onto second-order neurons located in the dorsal horn, which then send their axons into the contralateral anterolateral funiculus. Then they ascend as the lateral spinothalamic tract to the thalamus. Again, the ventral posterolateral nucleus we discussed before, and they terminate immediately behind the medial lemniscus. In the brainstem, the tract is located laterally and called spinal lemniscus, but joins the medial lemniscus at midbrain level, here. Finally, the primary sensory cortex in the postcentral gyrus is informed about the location and extent of a painful stimulus. Note that the signal is also relayed to other areas like the anterior cingulate gyrus, the prefrontal cortex, and to the insula for control and memory of painful stimuli. Nuclei of the reticulate formation, they are located in the brainstem, receive input from the ascending spinothalamic tract as well, which results in pain inhibition via the release of endogenous opioids in the spinal cord dorsal horn. Damage to the dorsal horn, for example at lower cervical levels, results in analgesia and thermal anesthesia, which means a total loss of pain and temperature sensation in this corresponding area. In addition, I would like to introduce you to the major pathways for transmission of unconscious proprioceptive information originating in muscles and tendons. They constantly inform the cerebellum about the position of joints and muscle strength to provide a fast regulatory mechanism to keep us from falling. The posterior spinal cerebellar tract originates in the posterior thoracic nucleus, called Clark's column, at the base of the dorsal horn in the thoracic spinal cord, and it's supplied by the trunk in the leg. The cunea cerebellar tract originates from the accessory cuneate nucleus, outside the cuneate nucleus in the medulla, for innervation of the ipsilateral arm. On the other hand, the anterior spinocerebellar tract monitors the state of activity of spinal reflex arcs and is partly crossed. It enters the cerebellum via the superior cerebellar peduncle, whereas the posterior spinocerebellar and cuneocerebellar tracts take the shorter route via the inferior peduncle. Both tracts mainly innervate the median and intermediate regions of the cerebellar vermis and paravermal areas. So in the next few minutes, I want to show you a plastic brain model of the human brain. It's comparable in size, and you are looking here at the brainstem that starts at the skull, as the foramen magnum, where the fiber tracts I just discussed in the spinal cord enter the medulla oblongata, or they descend from the brainstem downwards to the spinal cord. So here would be the transection site. This would be the start of the brainstem with the medulla, the pons, and here would be the cerebellum. So all three of them are called rhombencephalon. And above the pons, you see here the midbrain with the cerebral peduncles. This would be the pituitary gland. We will come back to that. So medulla, pons, midbrain, cerebellum. Now, if we want to have a closer look at the brainstem, we separate the two hemispheres. They are bilateral symmetric, and we will have a look at just one half, at a view that is called median sagittal. So here you see the hemispheres with the gyri and sulci. There's one prominent gyrus here. This is the cingulate gyrus that belongs to the limbic system. We will come back to that later. It is above the white matter commissure, the largest commissure in the brain, the corpus callosum, with a genu here and a splenium at the posterior side. So below the corpus callosum there is a glial lamella, the septum pellucidum. It separates the two lateral ventricles, and it's just above the fornix, a major fiber tract in the limbic system we will also discuss later. So the fornix is just above the roof of the third ventricle, which is located here. It's in the midline, and you see here at the roof the plexus choroideus, the choroid plexus that produces the cerebrospinal fluid. And here we have the thalamus, and here the hypothalamus, separated by the sulcus hypothalamicus here. This brown area is a connection between the two thalamic areas, the adhesio interthalamica. Here on the rostral side, you see the optic chiasm and the infundibulum that connects the hypothalamus to the hypophysis, the pituitary gland. In the posterior area, we have the epiphysis here and the posterior commissure here. There is the mammillary body here, belonging to the posterior hypothalamus. So the third ventricle is connected to the fourth ventricle here in the rhombencephalon via the aqueduct, this narrow canal connecting third and fourth ventricle. Behind the aqueduct, there is the tectum of the midbrain, and ventral to it, the tegmentum. Here in the midline, you see the oculomotor nerve exiting the midbrain medially from the cerebral peduncles that are ventral to the tegmentum. Here the oculomotor nerve, the third cranial nerve, exits in the interpeduncular fossa of the midbrain. Midbrain, pons, also the pons here bulges forward, and then medulla oblongata here, which will be connected to the spinal cord. So this would be the cerebellum with the vermis in the midline, exhibiting here the white matter branches that are surrounded by the gray matter, and this area is called the arbor vitae, or tree of life. So now if we now remove the hemispheres from the brainstem and the cerebellum, we turn the brainstem and have a look from the lateral side. Now, the thalamus here is belonging to the diencephalon, thalamus and hypothalamus. The diencephalon here is still connected to the telencephalic parts, the basal ganglia here, and the internal capsule fibers that are shown here, and laterally an older part of the cortex, the insular cortex, which is called the transitional or mesocortex here with gyri and sulci, the insula. Now, if we look here at the midbrain level from the lateral side, we see the cerebral peduncles ventrally, and dorsally the tectum with the colliculus superior involved in optical reflexes, and below that the colliculus inferior, which is part of the auditory system. And here we have the fourth cranial nerve, the trochlear nerve, that exits the brainstem dorsally, just below the inferior colliculus. Here we have the pons with the laterally located trigeminal nerve that divides into three branches: the ophthalmic, the maxillary, and the mandibular branch. And is located here laterally at the level of the pons. Also here between pons and medulla in the cerebellopontine angle, you find cranial nerves 7 and 8, the facial nerve and the sensory vestibulocochlear nerve. Here you see the cerebellar peduncle with the metal pin which connects the cerebellum here to the brainstem of this model. And below the pons you see the medulla from a lateral view with cranial nerves number 9, 10, and 11, the glossopharyngeal, the 9th, the vagus nerve, the 10th, and the accessory nerve, number 11. Now, if we move the brainstem and look ventrally from the ventral side, we have mesencephalon and oculomotor nerve, pons, the abducens nerve, the 6th cranial nerve, here exiting between pons and medulla. We have the hypoglossal nerve, exiting the medulla medially to the inferior olive nucleus, a very important motor nucleus belonging to the extrapyramidal system. And medially to the hypoglossal nerve, we have the pyramid here. The pyramid where the corticospinal fibers cross to the other side. So if we now put the brainstem back into the hemispheres, then becomes clear here that it fits here into the lumen of the lateral ventricles inside. And you realize that the hemispheres show the extension to the rostral side, to the occipital side, and this in this Widderhorn-like shape to the temporal part here. So here would be the temporal lobe, here would be the frontal lobe with the gyri and sulci, here would be the central sulcus, here would be the parietal lobe, and here is the occipital lobe. Now, if we now look at the temporal lobe in more detail, we will see at the bottom of the inferior horn of the lateral ventricle, we realize that there is a large structure called the hippocampal formation here. This is a very important part of the limbic system I will come back to later. So here you see the hippocampus, the dentate gyrus here, and the fimbria hippocampi, the axons that will form the fornix that we already saw below the corpus callosum when we looked from the median sagittal view. So the hippocampus is innervated by this region, the entorhinal cortex. Here, this is called the parahippocampal gyrus, where this cortex is located, and sends axons via the perforan path into the hippocampal formation. And finally, let's have a look at the cerebellum. So I already showed you the tree of life in the vermal region. So here would be the vermis. If you look at this median sagittal aspect of the cerebellum, here would be the cerebellar peduncles connecting the cerebellum to the brainstem. And the outer parts of the cerebellum are called the hemispheres. So the hemispheres that have an anterior part and a posterior part here of the cerebellum, separated by the primary fissure, fissura prima. And the cerebellum we will discuss in the next chapter. Let me introduce you to the cerebellum. A median sagittal schematic section through the brainstem reveals the cerebellar structure behind pons and fourth ventricle, the cerebellar vermis. And in the section shown here, it is referred to as the arbor vitae, the tree of life. Note that the folded surface of the cerebellar cortex contains fissures and foliae, unlike the cerebral cortex with its gyri and sulci. Some parts of the vermis are associated with the vestibulocerebellum, the phylogenetically oldest part of the cerebellum connected to the vestibular labyrinth. The anterior and paravermal parts developed strong connections with the spinal cord, following the requirements of quadrupedal locomotion in the first reptiles. The largest and newest developments, the cerebellar hemispheres, are mainly connected with the cerebral cortex. The hemispheres are the consequence of erect posture and achievement of various physical skills requiring special computing before complex behavioral programs can be formed in the motor and premotor areas of the cortex. 4 cerebellar nuclei are observed on each side of the cerebellum. The fastigial nucleus, connected with the vestibulocerebellar areas like the flocculus and nodulus of the vermis. The wrinkled dentate nucleus is associated with the ponto or neocerebellum, which conveys information from the cerebellar hemispheres to the ventrolateral nuclei of the contralateral thalamus. And the globose and emboliform nuclei, together called the interposed nucleus, which are relevant for control of posture and gait mediated by the spinocerebellar connections. In histological sections, the uniform structure of the tree of life can be appreciated. Innermost, the white matter, carrying the fibers that connect the cortex with the deep cerebellar and brainstem nuclei, followed by their granular layer, containing billions of small neurons, only 6 to 8 micrometers measuring in diameter. The axons of the granucells divide in a T-shaped manner inside the outer molecular layer, which is below the peel surface, and they form the parallel fibers which excite the Purkinje cells of the cerebellar cortex, which are situated just between the molecular and the granule layers, but they are not visible here in this magnification. The Purkinje cells are the inhibitory output cells of the cerebellar cortex. They do not project out of the cerebellum, but to the deep cerebellar nuclei. Here you see a transverse section through the midbrain, which is divided into 3 principal regions. The tegmentum in the intermediate position, which extends the tegmentum of the pons and medulla, and ventrally the peduncular region with the 2 large crura cerebri and a dorsally located tectile area behind the level of the aqueduct of Sylvius. This narrow canal allows cerebrospinal fluid to flow between the forebrain and the brainstem. Just ventrally to it, the nuclei of the oculomotor nerve, the 3rd cranial nerve, are shown, as well as the paired red nucleus, which is so named because of its high iron content. As we will see later, the red nucleus and the substantia nigra play major roles in the so-called extrapyramidal motor system. The cerebral peduncles carry corticopontine and corticospinal fibers projecting caudally to pons and spinal cord. They originate in the cerebral cortex of all telencephalic lobes and follow a course through large subcortical nuclei, the caudate and the putamen, which together form the striated body, the corpus striatum. These caudally projecting fiber bundles plus fibers connecting the thalamus with various cortical areas form the internal capsule, which passes between the thalamus and the basal ganglia shown here. Fibers in the retrolentiform and sublentiform parts of the internal capsule are derived from the occipital and temporal lobes. The anterior limb of the internal capsule carries fibers from and to the frontal lobe. The midbrain is also the most relevant source of an important neurotransmitter, dopamine. It is the pars compacta of the substantia nigra, which plays a key role in the control of movements by innervating the striatum. The death of the dopaminergic neurons located here causes Parkinson's disease. In addition, dopamine originating from neurons located in the ventral tegmental area of the midbrain is also a transmitter in the so-called mesocorticolimbic system, implicated in drug and natural reward circuitry. Major projections of the VTA reach the nucleus accumbens and the frontal lobe. The ventral tegmental area is involved in cognition, motivation, drug addiction, and several other psychiatric disorders. To better understand the development of the basal ganglia, it is useful to have a look at the frontal schematic section through the developing and through the adult brain. They reveal the importance of the internal capsule in creating the final anatomy not only of the basal ganglia, but also of the diencephalic nuclei, which are partially pushed laterally by the caudally projecting fiber bundles. In the upper part, you see the telencephalic vesicles with the lateral ventricles, the choroid plexus of the lateral and the third ventricle. These produce the cerebrospinal fluid. And you see the telencephalic ganglionic eminence. It is the source of the subcortical nuclei, including the basal ganglia. In early stages, the third diencephalic vesicle wall can be divided into 4 parts. The dorsally located posterior part, the epithalamus, giving rise to the pineal gland, smaller nuclei, and the posterior commissure. Then the major part, the dorsal thalamus, which in most textbooks is simply called the thalamus. It is the largest nuclear mass in the entire nervous system and contains specific nuclei projecting to identified regions of the cerebral cortex and non-specific nuclei that do not correspond to any one sensory modality. The metathalamic nuclei, the medial and lateral geniculate bodies, are located at the back of the thalamus and are connected with the primary auditory and visual cortices, respectively. The main components of the subthalamic mass are the globus pallidus, which receives its input mainly from the striatum and the substantia nigra. It projects to the ventrothalamus and to the other component, the subthalamic nucleus, as well as to the midbrain. All of these nuclei are involved in generating motor activities. The subthalamic nucleus is very important for suppression of unwanted movements and strongly connected to the medial pallidal segment. The most ventral part of the diencephalic vesicle is the hypothalamus, a major control center for all autonomic and endocrine functions via its regulation of hormone release by the pituitary gland, the hypophysis. The hypothalamus controls body temperature, appetite, thirst, circadian rhythms, and many aspects of reproduction, parenting, and attachment behaviors. The mammillary bodies listed here are additionally involved in learning and memory, as we will see later. They receive most of their input via the fornix, a major pathway of the limbic system that we will also discuss later. It is located just below the corpus callosum, the major commissure connecting the 2 hemispheres. Here you see the final locations of the caudate nucleus and the putamen, the structures derived from the ganglionic eminence of the telencephalon, as well as the diencephalic nuclei, the pallidum, globus pallidus, which is pushed apart by the internal capsule, and putamen and globus pallidus together form the lentiform nucleus. But note that they are derived from different sources. Also shown are the thalamus and the hypothalamus, with the optic tract as landmark and in the midline the third ventricle. In transverse sections through the adult brain, we can identify the following structures. The thalamus derived from the diencephalon, the caudate nucleus derived from the ganglionic eminence, the lateral ventricle just medial to it, the fornix in the midline, the hippocampal formation in the temporal lobe bordering at the inferior horn of the lateral ventricle, and the splenium corporis callosi. Note the highly folded neocortex with the white matter shown here. In a section just below, we find the basal ganglia, medial from a gray lamella, located between the insular cortex and the putamen, which is called the claustrum. Also visible are the fibers of the internal capsule. the hippocampus, the fornix, and the third ventricle. The hippocampus and its major output fiber tract, the fornix, lead us to the next chapter, the limbic system, which is derived from phylogenetically older cortical structures, the archae and the paleocortex. Broca, over 150 years ago, coined the term limbic, indicating a limbus or rim of cortex around the corpus callosum, the cingulate gyrus, which is located above the corpus callosum here. The insular, parahippocampal, and cingulate gyrus comprise the outer limbic system, while the structures shown here mainly constitute the inner limbic system. This is a sagittal schematic view with a few landmarks for orientation. The hypophysis is shown, which is connected to the diencephalon by the infundibulum. Pons, optic chiasm, and genu and splenium of the corpus callosum are indicated as well. The phylogenetically old archicortex gives rise to the hippocampal formation. It includes the pyramidal neurons of the cornu ammonis and the dentate gyrus. Most of the efferent fibers of the hippocampus exit this key structure involved in learning and memory via the fimbria and travel in the crus, corpus, and columnar fornices to the mammillary bodies located in the posterior hypothalamus. Axons also cross the midline in the commissure of the fornix and innervate the contralateral limbic lobe. From the mammillary bodies, the signal is synaptically relayed to the anterior thalamus via the mammillothalamic tract, and this thalamic nucleus in turn is strongly connected with the cingulate gyrus, which completes the main limbic network named after Papez, the Papez circuit. The Papez circuit is driven by the perforant path originating in the entorhinal cortex, shown here in the anterior part of the parahippocampal gyrus. The section also reveals the large amygdaloid complex, just anterior of the hippocampal formation in the frontal part of the temporal lobe. The amygdala belongs to the limbic system and is primarily associated with the emotion of fear. The insula, the thalamus, and the basal ganglia are shown as well. Note that this frontal section clearly shows the cingulate gyrus, right above the corpus callosum. Ramon y Cajal's beautiful drawings of the hippocampus reveal the structure of the archicortex. The silver stain technique developed by Camillo Golgi allows the reconstruction of the morphological details of the principal cells located in the hippocampal Ammons horn. The pyramidal neurons with their long apical dendrites exhibiting hundreds of spines and serving as the postsynaptic site for incoming activity derived from the perforant path. The axons are collected in the fimbria and travel via the fornix to the mammillary bodies and to other nuclei of the limbic system. Limbic structures also include the cholinergic centers located in the basal forebrain, such as the medial septal nuclei and the basal nucleus of Meinert, which supply the neocortex with the neurotransmitter acetylcholine. Moreover, brainstem nuclei located in pons and mesencephalon provide cholinergic innervation to various areas of the diencephalonic limbic system. The limbic system, generating memories, emotions, and several key aspects of our life, is under control of the highest level in the hierarchy of nervous structures, that is the neocortex. In the final few slides, I want to introduce you to this newest and most complicated part of our brain. Let us start with its histological features. Particularly striking is the cytoarchitectonic homogeneity of the neocortex, which is suggestive of a functional regularity. It is now clear that the main organizational unit of the neocortex is the cortical column, a group of neurons that can be successively penetrated by a probe inserted perpendicularly to the cortical surface and which has nearly identical receptive fields. In routinely stained histological sections, such as this one, cortical columns cannot be identified, Instead, a laminar structure becomes somewhat apparent. The exact borders of the 6 different laminae are not easily discernible, but in general we differentiate. Number 1, the molecular layer, mostly filled with neurites and apical dendrites derived from large pyramidal neurons in layers 3 and 5. Number 2, the outer granular layer, containing small pyramidal and stellate interneuronal cells. Number 3, the outer pyramidal layer with many medium-sized pyramidal cells. And number 4, the inner granular layer, comprised of stellate cells receiving afferents from thalamic relay nuclei. They are especially numerous in sensory areas, in the so-called granular cortex, but fewer in motor areas, the agranular cortex. Number 5, then, the inner pyramidal layer contains large pyramidal cells projecting to the striatum, brainstem, and to spinal cord. Finally, number 6, the fusiform layer, contains modified pyramidal cells projecting to the thalamus. Bordering lamina 6, you see the white matter, containing mainly myelinated axons. The histological and cytoarchitectural differences are the basis of the neocortical reference map introduced by the German neuroanatomist Corbinian Brodmann over 100 years ago. He labeled the motor areas with numbers 4, the primary motor cortex, 6, the premotor cortex, and 8, for example, the frontal eye field. The primary sensory cortex, S1, is contained in areas number 1 to 3. Area 17 is the primary visual cortex, with areas 18 and 19 representing the visual association cortex. Areas 41 and 42, for example, represent the primary auditory cortex. Area 22, the auditory association cortex, and so on. Before Brodmann came up with the numbering of cortical areas, anatomists named the cortical gyri and sulci to indicate different surface regions. Since the human cortex is folded, they marked the grooves, here drawn as black lines, as sulci. These surround the different gyri. For example, in the frontal lobe, the major gyri are named the superior, medial, and inferior gyrus. The lateral sulcus separates the frontal lobe from the temporal lobe, here shown in green. And the central sulcus separates the frontal from the parietal lobe. The occipital lobe is shown in blue here. Since this tutorial is intended as basic course for students and colleagues interested in the Human Brain Project, and probably with a background in math or informatics, I want to finish my presentation with some quantitative anatomical data on neuron numbers and connectivity in the neocortex. First of all, it is surprising how similar the basic structure of the neocortex is between species, considering the major differences in cognitive abilities. For example, most species have 50,000 to 100,000 neurons under 1 square millimeter of cortical surface. The cortical thickness varies a lot and is highest in humans. 100 million cortical minicolumns with up to 110 neurons each are found. And cortical neurons are packed at really high densities, 100,000 neurons per cubic millimeter in the mouse. Humans have 24,000, rats 55,000, and 6,000 in elephants. Let us compare the 3 different vertebrate species, the elephant, the man, and the rat. With 4,800 grams, the elephant clearly has the heaviest brain, also the largest neocortical area with 630,000 square millimeter, but the lowest density of neurons, 6,000 per cubic millimeter. Rats exhibit comparably high neuron densities of up to 55,000. The values for a human brain are in between. 1,300 grams, 250,000 square millimeter surface area, 24,000 neurons per cubic millimeter. But taking into account the cortical thickness, we end up with the following absolute number of neurons. That is the number of operational units in our neocortex. And this is highest in humans. So the human neocortex contains 20 billion neurons, nearly twice as much as the elephant, and 1,000 times more than the rat. Interestingly, the cortex of cetaceans, whales or dolphins, contains even more neurons, up to 37 billions. However, recent work on dolphin behavior does not support the hypothesis that these animals exhibit cognitive abilities which are superior to humans or even to monkeys. So it is not clear what purpose these high neuron numbers serve in dolphins. Finally, a few more numbers obtained with the electron microscope. Each neuron contains 500 other neurons on average. Synaptic density is remarkably similar at around 1 billion synapses per cubic millimeter. And 80% of the cortical neurons are excitatory, 20% inhibitory. The average number of synapses per neuron is 8,000. And dependent on cortical thickness, the axonal length is approximately 4 kilometers per cubic millimeter and dendritic length 0.4 kilometers per cubic millimeter. So taken together, we may conclude from these data that the neocortex is concerned with the most abstract level of sensory analysis and behavioral guidance. It has wide access to information from nearly all receptor systems, allowing it to couple complex behavior to a variety of sensory events. Due to massive interconnections, the cortex is able to correlate a large variety of stimuli, regardless of the modality involved. Interestingly, a consistent pattern across all areas of the neocortex is that most of the thousands of synapses formed on the dendritic tree of any neuron come from its neighboring excitatory neurons, located in the same cortical area. Only few synapses are contributed by long-distance connections, whether they arise from neurons in the thalamus or in other cortical areas. Thus, the local circuit is at the heart of cortical computation. Clearly, the building blocks of our neuronal networks, neurons with all their processes, the axons and dendrites, evolved under tight constraints of energy consumption and necessity for speed. So the brain consumes only 17 watts of energy, while a silicon version of the brain would use up to 10 megawatts. It would simply melt. This is the reason why our brain uses significantly slower computational elements to solve problems that much faster computer hardware cannot achieve. However, the neocortex is a fast computer. Consider the speed of face recognition performed in our visual associative cortex areas when we identify a familiar face out of several thousand images stored in our brain. This takes usually longer for our PC at home, since digital images are compared on a pixel-to-pixel basis, which the brain does not. So understanding the structural basis of these amazing abilities of the human brain, this remains a subject of tremendous fascination for neuroscientists and colleagues in other scientific fields. By applying advanced neuromorphological and high-resolution imaging techniques, we are able to analyze structure-function relationships of highly complex neuronal systems. Let me finish with a quote by Gerald Edelman, an American biologist who once said, "If someone held a gun to my head and threatened oblivion, if I did not identify the single word most significant for understanding the brain, I would say neuroanatomy." Indeed, perhaps the most important general observation that can be made about the brain is that its anatomy is the most important thing about it. End of quote. And with this remarkable insight of the 1972 Nobel Prize winner in Physiology or Medicine, I thank you very much for your attention.